So this is day 3 of our EOC review. And what we're going to look at first is the double-stranded and complementary nature of DNA. Let's remind ourselves of a couple things before we get started here. DNA, what does that stand for? Let's remember that it stands for deoxyribonucleic acid. Or I'm sorry, deoxyribose nucleic acid. When you're looking at a molecule of DNA or a strand of DNA, you do need to remember it's double-stranded. In this specific picture, this is one strand of the DNA molecule, not two strands. So it says, below is a strand of DNA. DNA in the cells exist in a double helix. That is the shape of our DNA molecule. You should remember it looks like a twisted ladder. Double helix. What else needs to be added to this strand to make a double helix? Give the nucleotide sequence. So what we're looking at here is a single strand of nucleic acids or nucleotides. Couple things we need to think about first is that a nucleotide is the name of the monomer or the subunit of a DNA molecule. In this strand, the strand is actually lying sideways. You have 3 parts to a monomer or to a nucleotide of DNA. The first part right here, it says PO4. Do you remember what that stands for? What element that stands for? That's our actually phosphate group. Notice there is an oxygen attached to that. Next, you have this little pentagon structure, and that actually stands for our sugar, which is also known as deoxyribose. That's our second structure, or second part of our nucleotide. And then the last thing right here, these are the parts that will vary, and this is going to be our nitrogen base. And in DNA, you have 4 types of nitrogen bases, and you can actually find them here. That's going to be A with T and C with G. Notice how I'm writing that. They do pair up in a very specific way. So when we talk about DNA being complementary, that means they have very specific ways of pairing up. Okay. So again, this is only one side of our DNA double helix. And so what they're asking us in this first question here, what else needs to be added for this strand to make it a double helix? Well, we need the second side of that. And so they're giving us the nitrogen bases of one strand, and that's how we name the nucleotides. And they want us to name the next strand. So if we start at the top, or actually I guess we could start on this side, G will pair with what nucleotide, or which nitrogen base? G is always going to pair with C. How can we write this? I guess I can just write a little dash here. G will pair with C. A will pair with T. C will pair with G. C again will pair with G. G will pair with C. And A will pair with T. However, between these nitrogen bases, what is going to connect them? What type of bond is that? That is going to be a weak, and maybe you've guessed it by now, hydrogen bond. It does have to be weak to allow for protein synthesis and DNA replication to occur. So here in these questions, what are the black pentagons? Again, we said this was the sugar molecule. And where are the nitrogen bases? Right down here. Okay, and one thing we'll want to add to that is the nitrogen bases only varying part. So that's the only part of the nucleotide or only part of the DNA structure that's going to be different. Next, we do want to compare RNA and DNA in the following table. Remember that RNA is a little different because it is single-stranded. So compare RNA and DNA in the following table. First of all, in RNA, we do have a different sugar. We already said in DNA that it's deoxyribose. In RNA, it's just called ribose. In RNA, we have A, T, C. I'm sorry, we don't actually have a T. We have A and a U pairing up, and then C and G. I'm going to fill in the RNA side. I want you to fill in the DNA side. Number of strands, RNA has only one strand. Where is it found in the cell? It's actually found, if you think about protein synthesis, it can start in the nucleus and go to the cytoplasm. Because there are actually different types of RNA. The main function, however, of RNA is going to be help make the molecule that does all the work in our cell, which are called proteins. On the other hand here, DNA is going to code for proteins. The instructions for everything that happens inside of our cell. Go ahead and make sure you fill in the bases for DNA, the strands, the number of strands in the DNA, and where can we find the DNA. Remember, DNA is very important. It has to be in a protected area so it does not get damaged. Next, what kind of weak bond holds the two strands of DNA together? We just answered that above, so you should be able to fill that in. And why is it important that these bonds are weak? Again, I just mentioned them above for two specific types of jobs that the DNA needs to do. So you want to make sure you'll rewind a little and figure out that answer. We will go ahead and move on to the next little section. Make sure you fill in the few blanks that I've left for you. And the next part, we're going to explain how DNA and RNA code for proteins and determine the traits. So if the strand of DNA above, the one above, undergoes transcription, what will the sequence of mRNA be? So we're talking about the strand that we initially gave you, and we'll start from the left. Initially, the strand above had G, A, C, C, G, A. That was the strand of nucleotides you had in your drawing. What we're going to do is, what will the sequence of mRNA be? Remember that when we move into mRNA, this is called transcription. So we'll circle this right here. And that means we're going from DNA to mRNA. And do you guys remember how that base pairs? Well, G always, always, always pairs with C. A usually pairs with T in DNA, but in mRNA that's different. Instead of having a T, we have a U for uracil. C will pair with G, C again will pair with G, and G will pair with C, and that last A will pair with the U. Then it says, after translation, what would the amino acid sequence be for this section of mRNA? Read from right to left. We've already started from left. To right, so let's go ahead and change that. Left to right. So we already have our mRNA. We want to figure out what would our amino acid sequence be. Remember that the letters above here, the words here, are the amino acids, which basically is the monomer of proteins. So the first thing we have is CUG. This is our first three letters. Here you have your C, first letter, U, second letter, G, third letter. So that's going to be L-E-U, which stands for leucine. Then we're going to have G-C-U. Here we have our G, here we have our C, and our U. So our amino acid is going to be ALA, A-L-A, alanine. So these two words would actually make up the chain of amino acids, or the chain for the protein that we're trying to make in our cell. But the next question does ask us, what is a codon? And a codon is a set of 3 nitrogen bases in mRNA that code for an amino acid. So in this chain that we originally had, how many amino acids do we code for? We actually code for 2. So here's one codon. Here's our second codon. And then it says what kind of bonds hold the amino acids together in the protein that is formed. Now a protein is a polymer of amino acids. And the type of bond that holds it together, we've seen once before, is a polypeptide. Next we have the question, what are 3 types of RNA and what are their functions? This is going to be mRNA, tRNA, and rRNA. I want you to fill in the functions of those 3. Next, it does say, how does the protein code for traits? Remember the alien activity? How does it code for traits? And you want to go and think about that. The next thing I want you to do is I also want you to fill in these 3 questions right here at the bottom. And after you're done with that, you should finish up your summary sentences. And that's going to be all for this afternoon.